Okay, good afternoon, everybody. My name is Monray Williams. I'm from the University of the Western Cape in Cape Town, South Africa. And today I'll present my research I've conducted over the past 2 years, which is titled The Molecular Validation of Putative Antimicrobial Peptides for Improved HIV Diagnostics via the protein P24. So the disadvantage of presenting so late in the day is that you're possibly going to hear something that you already been hearing throughout the day. But just as a brief introduction, we know that HIV and AIDS is a disease of the immune system. The common methods of spread is through unprotected sex, transfusion of infected blood samples, sharing of contaminated equipment, or from mother to child. Now, we know that HIV functions by attacking T helper cells. Now, these cells are known as very important cells within the immune system as they're responsible for various pathways such as the production of antibodies or activating pathways such as the cytotoxic T lymphocyte pathways. And obviously with a reduction in these cells, there's basically a reduction in the immunity, and this provides a gateway for secondary infections to come into play, such as TB. Now, HIV has a very unique structure, which also attributes to its unique mode of action. It's also virulent ability or capabilities, and it's also its high stats related to it. So by the year 2014, 35 million people were infected with the HIV virus. To bring this closer to home, where I'm from, sub-Saharan Africa, 25.8 million people were known to have the virus. Swaziland having the highest prevalent rate of 26.5%, and South Africa is known to have the world's largest HIV population. Now, this can be attributed to various things, but we can mainly blame the poor therapeutic system that's not where we want it to be, and also by the poor diagnostic systems. Now I'm going to use this diagram to depict or kind of explain the various diagnostic systems we have that we heard of today also in earlier presentations. Now, there's two methods of detection. We can either detect it by detection of the antibodies or detection of the actual viral particles itself. Now, the most common one is the antibody testing, where you use the rapid test, for example. This is an advantage or great advantage for implementing in resource-limiting areas because results can be given quickly. However, the main disadvantage is that you often have to wait for a window period for a successful test, which can be anything from 4 weeks post-infection up until 3 months. So the goal of diagnostics is always to be able to accurately give results as early as possible, so the person can become educated, and so the HIV infection can kind of be reduced by making the correct life decisions or adaptations. Now, an advantage of this is what we've seen earlier is the RNA test. It's a great test, it's accurate, it's one of the most sensitive tests. However, it's considered as something that's very expensive. It requires specialized staff and equipment, and it's something that's difficult to implement in resource-limiting areas such as Africa or the developing countries. So we often look at the P24 antigen assay as a slight improvement. It's able to decrease the window period. I'm going to just dwell or go into it a bit more in detail because this is the assay that I'm currently looking at. This assay can decrease the window period as mentioned, however, it's considered as insensitive. It's insensitive that it's placed in 50% of asymptomatic patients false negatives. Now, the reason for this is that soon after infection, the P24 antigen presents itself. You have the P24 antibody, which would bind to the C-terminal domain of that P24 antigen. Now, the current detection system uses an antibody that targets the same epitope to which that P24 antibody is already bound. So if that epitope is saturated by the P24 antibody, the detection antibody won't be able to pick it up. So current work that's being done is they're actually trying to separate the complex itself so that they then can detect the P24 antigen. Now, again, this requires an additional step, which is known as a booster step, which is either by adding a certain chemical or by a heating step. And again, this makes it difficult to implement in resource-limiting areas where sufficient science or clinicians can perform these experiments. The additional approach that could be taken is the one that we decided to go with, is to use a molecule that targets another epitope on the P24 antigens. And therefore, we predict that whether that P24 antibody binds or not, we would be able to target another epitope on that antigen and then still be able to detect the P24 antigen within a patient sample. So we identified antimicrobial peptides as a possible solution to this, or AMPs. Now, the reason we looked at AMPs is they are part of the innate immune response. They're found in prokaryotes and eukaryotes, generally small positively charged amino acid amphiphilic molecules. And they have shown to be a broad spectrum of activity from gram-positive, gram-negative bacteria, protozoa, fungi, and as well as viruses, more specifically in our case, HIV viruses. It has been shown, according to literature, that it's very difficult for a microbe to develop resistance against AMPs due to the diversity and the unique mode of action. So basically, a microbe would be required to redesign its outer membrane to resist the effects of an AMP, and this is often considered as a chemical or an energy-expensive process for most microbes. Thus, we're looking at using a peptide or a peptide over antibody approach to do the detection. Now, the reason is for this: peptides are small, rapid and reproducible synthesis, simple and controllable, less labor-intensive than using antibodies. We have a broader spectrum or scope to work with, high stability, non-toxic. It lacks immunogenicity, so working with antibodies could be things like non-specific binding to target molecules, time-consuming, and we often have poor linearity of dilution. So this brings me to the methodology. Basically, my research was done as a bigger project with another member within the Nanotechnology Innovation Research Group. So basically, he identified AMPs or antimicrobial peptides which binds to the N-terminal domain. So I'm just going to repeat. If the P24 antibody binds to the C-terminal domain and we identified antimicrobial peptides which binds to an additional pocket, we predicted that these AMPs will still be able to detect the P24 antigen within a patient sample. So how we did this, he uses a mathematical algorithm called HOMA. And then we created a model, and the model was used to scan various protein databases. And then this created a list of novel antimicrobial peptides which were predicted to have anti-HIV activity. Now, this anti-HIV activity was based on its binding to target molecules. And similarly, we wanted to exploit that binding to target molecules in a diagnostic approach. So we basically used the top 10 AMPs with the lowest E-value, and then we performed an in silico or bioinformatic approach first, where we predicted the 3D structures of these antimicrobial peptides, and then performed a docking study or protein-protein interaction study between the AMPs and the protein P24 antigen, and then to do visualization by a software called PyMol, and then we identified the 10 AMPs which binds to an additional pocket of the P24 antigen and then might be able to be used in an improved diagnostic kit. So this brings me to the aims of my study. My aims was to create derivative AMPs, which shows higher predicted binding affinity for the N-terminal domain of the P24 antigen, then to molecular validate its binding to the P24 protein, and then once we identified the best suited AMPs for this, is to create a prototype that uses AMPs and conjugate it to gold nanoparticles, and then use it to detect HIV within patient samples. So my methodology, we started with the in silico approach, or bioinformatic approach. The bioinformatic approach was to identify derivative AMPs as mentioned. So we basically looked at the AMP sequences that we had, and we subject to a server to identify hotspot residues. Now, hotspot residues are residues that we know between the complex of the P24 and the AMP, those amino acids in that complex that contribute to most of the binding free or the binding energy. So we knew that those amino acids would be sensitive to mutation. Then basically we formed an in silico site directed mutagenesis. We predicted the 3D structures of the derivative AMPs, deep patch dock server or protein-protein interaction studies of these derivative AMPs, and then visualize and interpret results by PyMol. After this, we did a molecular approach, where we synthesized all peptides, derivative and parent peptides, and then we purchased a construct containing the P24 antigen sequence, and then we basically used a PGX vector to express the P24 protein within our lab, and then we used an in-house binding study where we used AMP combinations, either conjugated the gold nanoparticles to detect the P24 antigen that we expressed in the lab. So we use those AMPs in combination with each other on the lateral flow device, and then we use it to detect purely the P24 antigen. Once this was determined, we were able to see which AMP combination best detected that P24 antigen, and then we implemented that in a prototype to detect HIV or look at HIV positive samples. So to bring it to my results, first we start with the in silico results. All mutations that we introduced were of similar characteristic to the parent amino acid. Okay, it was either to introduce amino acid with a higher net positive charge or greater side chain, which will I explain in the coming slide. We look at the physicochemical properties just to confirm that these antimicrobial peptides are still AMPs. We looked at certain amino acids, the presence thereof, amino acids such as positively charged amino acids, as shown in literature that AMPs function better with a higher net positive charge or a higher presence of these amino acids. Cysteine was also included as they were known to help with folding of proteins and thus better folding into pockets and thus better binding activity. Again, the presence of these amino acids had a direct link to its total net charge, and also looked at things like the Bowman index and the instability index. Now, a Bowman index is just a protein's binding potential. It's basically a score of less than 2.5 indicates that a protein would have a more specific binding to a target, where instead of have a hormonal type of binding where it can bind various targets. The instability index is basically just the stability of the peptide doing certain studies. This is the 3D structure prediction of the AMPs. We can see that there hasn't been a great change in the structure itself. However, if we go to the next slide, we can compare the docking studies or the protein-protein interaction studies, where we can see that when we did the mutations, that all AMPs had an increase in binding affinity by at least 4.2%. Now, AMP1 and AMP8 becomes very important because AMP1 show the highest binding affinity, or the derivative shows the highest binding affinity, where AMP8 had the highest increase in binding affinity. So this is just an image depicting the P24 antigen and the change in binding or the shift in the binding pocket. We can see that the AMPs still remain at the N-terminal domain and thus are still predicted to have to be able to be used in an improved diagnostic kit. This is the results of the P24 protein expression, where we use the GST purification system to successfully express the P24 protein at the base pairs of 223 kilodalton. This is the results of the protein interaction study. So basically what we did, I'll just re-explain it, that we put certain AMPs combinations with either in the conjugate pad of the lateral flow device or the membrane side of the lateral flow device, and then we used our P24 protein that we expressed in the lab. We lyophilized that sample and then resuspended in certain buffers, and then ran it on the specific kits. So basically we had a G rating. G rating is the gold standard, which is indicated by the intensity, which is indicated in the lateral flow devices window. So it's a rating from G1 to G10. We can see that AMP1 gave a G8, where its derivative form gave a score of G10. So we can see that the derivative forms that was predicted in silico did in fact give a better binding. So once we knew that these two AMPs here gave the best binding affinity, we then implemented that in a lateral flow device, and that is what you see on the right-hand side there. We can see that if we use a negative P24 sample, there's no line indicated at the test line. Obviously, this was expected because there was no P24 antigen present in that sample, and then a slight band indicated by the test line of using a positive P24 sample, which is the one we expressed in the lab. So we knew that these two AMPs were the best suited ones, and then these ones we implemented in a lateral flow device prototype to detect HIV samples. So what we did was to use global HIV standards. We started by using a negative HIV sample. As you can see, that there's an indication of no test line. This was expected because we believe that there's obviously no P24 antigen present in that sample. Then we just retested the P24 protein that we expressed in the lab. You can see a slight band at the test line there. And then we test global HIV standards for HIV 1 year and HIV 2 year. So we see that we're able to detect both, and great band intensities were also indicated. Now what we realized afterwards is the possible reason for the low band intensity displayed for the P24 antigen is that we loaded a concentrated sample of 100 micrograms per ml. And doing further literature reading, we realized that the mean concentration of the P24 sample is 200 picogram per ml. Now, what has been happening here is known as the hook effect in lateral flow devices, where unconjugated gold nanoparticle sample already migrates down the lateral flow device and binds to the test line. So without the kind of say a fluorophore or indication, these molecules would bind to the test line, and then thus a lower concentration is indicated on the test line. So this is definitely something that has to be redone in future, just to confirm that it is able to detect at the same concentration as what it was in the patient samples or the global standards. So in conclusion, with the help of bioinformatics, we are able to identify novel antimicrobial peptides, which were implemented in a lateral flow device, which accurately detects HIV 1 and 2. Our future work could be maybe using another biophysical technique to confirm the binding, such as surface plasmon resonance, between the AMPs and the P24 protein antigen, and then also to look at to elucidate the structural binding interaction by looking at NMR. We then want to look at the field study of testing at least 500 patient samples on these lateral flow devices, but also more specifically to look at different stages of infection so that we know that it's able to detect across various stages of infection. And then, as mentioned, these AMPs were predicted to have anti-HIV activity. So this is currently work that is ongoing within our research group. And that's the references. And then just acknowledging my supervisors, the Nanotechnology Innovation Center, MINTEC, National Research Foundation, and Medical Diagnostics, Mr. Ashley Ace, which help with the lateral flow device or the commercialization or the testing of those samples. Okay, thank you for your time.